Good morning, my name is Blanca Quintero and I'm PhD student at the University of Seville, Spain. I wish to thank the organizer for the opportunity to participate in this interesting meeting. In this talk, I will describe briefly some interesting studies from our laboratory about the strategy and brain mechanism for spatial cognition in teleost fish. Most studies on spatial cognition have traditionally focused on mammals and birds, which are usually considered the most advanced vertebrates. In contrast, teleost fish have been traditionally considered primitive vertebrates in regard to brain complexity and behavior function. However, an increasing of evidence indicate that teleost brain is comparable with the brain of land vertebrates, and that teleost fish also show advanced cognitive capability, including spatial navigation skill that has parallel those of land vertebrates. So the brain of teleost fish is comparable to the brain of land vertebrates. In regard to spatial learning, fish like land vertebrates are able to orient or navigate using several cognitive mechanisms based on distinct and parallel spatial learning and memory systems. In fact, we know now that many of sophisticated cognitive capabilities that have been supposed exclusive of mammal and bird, for example, allocentric navigation or cognitive mapping, are also present in fish. We know that cognitive mapping strategy depend on the hippocampus in mammals and birds. Comparative neuroanatomical and functional evidence suggests that the telencephalic region homologous to the hippocampus of land vertebrate is present also in teleost fish. We can see here that teleost fish have a same structure homologous of hippocampus as birds, reptiles, or mammals. However, we know that it exists two types of embryonic development of vertebrate telencephalon. We know that the brain of non-anterior region have a evagination process. In contrast, that's the process of eversion in aptenopterygian. This difference in the embryonic development also produced the reversal of the paleotopography observed in different area of telencephalon, thus the hippocampal pallium, that seem to be placed in a lateral position in telencephalic hemisphere in aptenopterygian. Although there is an incomplete agreement about the function of the hippocampus, an important amount of studies indicate that in mammals and birds, hippocampus is important for different processes of and type of memory. For instance, spatial memory, place learning, allocentric navigation, cognitive mapping, relational memory, or trace memory, but not for non-spatial discrimination, cue learning, egocentric orientation, taxon strategy, individual memory, and non-trace memory. As I said before, in this talk I will detail some work that have helped to study the strategy and mechanism of a spatial memory system in teleost fish. The first experiment that I want to talk about is an experiment did by Rodriguez in 1994. Goldfish was training in three different procedures that had used a allocentric and egocentric strategy to find a goal in a plus maze. The place procedure implicate, im, im, implicate that the information provided by the distal cues based on cognitive maps. So for the place training, the fishes develop an allocentric strategy. So in the place group, two different start positions were used at the random, but the goal was situated always in the same place in the room. Thus, animals in this group could only use array of extra maze cues to solve the task. So no egocentric strategy were adequate to reach the goal. In contrast, for the turn procedure that use egocentric strategy provided by a particular turn response have had two different start position for two random position, but in every trial, the goal arm was determined by a fixed turn response. In this case, in this graph, the turn response had always turn right in this group of animals. And also, we use two type of transfer test. So we do the transfer type 1 that we only change the start position of the animal, but the maze in the same position in the room that the animal was trained. Here we can see that the animal trained by place procedure always uh, uh, um, always are able to reach the goal in the same position that is was training. But the animal that was trained in a turn procedure always did the turn response that they learned. For example, in this case, turn right. For the transfer test 2, the start position also is changed, but we change the position of the maze in the room, and we see that only the group of was training in the place procedure with allocentric strategy are able to reach the goal in the same position that this was training. In the case of the turn procedure, or the animal that was training in a turn procedure, always start in the one position on the other position, make the turn response that is was learn. For example, in this case, turn right. For different, also there was proof with a totally or partial occlusion of the different cues in the room. When we see that the animal that training in a place procedure and we occlude all the cues, we see that the animal is was disorientated, and this animal didn't able to reach the goal that it was training. But when we only occlude partially this cue, the animal can complete all the relationship of the different cues in the room, and they can reach the different goal that it was training. With the study conducted by Salas in 1996, we can see that the telencephalon have an important role in the allocentric strategy in navigation. We can see that the animal with a complete ablation of telencephalon have an important domain in allocentric strategy, but not in a mixed procedure or egocentric procedure. So, the telencephalon ablation produce a remarkable spatial cognitional impairment in the group of that was trained in allocentric with allocentric strategy, so in a place procedure. Here we can see a representation of one brain in a sham group in a ablation of telencephalon group. Also, with the work of Rodriguez, we can see that dorsolateral telencephalon lesion in goldfish produce significant deficit in spatial navigation tasks that require the use of map-like strategy. So, we can see that the lateral pallium lesion can produce an impairment as severe as that observed in the animal with complete ablation of telencephalon. Moreover, none of the pallium lesions produce deficit in cue learning. These results indicate that the lateral pallium of teleost fish, but not the medial or dorsal pallium, plays an essential role in the processing and encoding of spatial information by means allocentric memory representation. Also, Duran, using different behavior procedures, obtained convergent results regarding the involvement of the goldfish hippocampal pallium homologue in map-like spatial navigation. We can see that the lesion of DL have important involvement in the codification of the proximal cues. When we remove the proximal cue, we can see that the animal with a lesion in DLB can do as well as the sham group of the lesioned animal. So this result demonstrated that the dorsolateral telencephalon rely exclusively on the individual local cues in the immediately vicinity of the goal placed to solve the task, and not on the complete array of landmarks. So it's important that these results closely resemble that obtained after hippocampus lesion in mammal, birds, and reptiles, and demonstrated that the dorsolateral telencephalon on teleosteofish is a critical brain center for allocentric navigation based in map-like strategy or map-like spatial memory. Also, there exists some works as Duran that show that cerebellum also have a role in the solution of the whole board task. The lesion of telencephalum produce a profound spatial cognition deficit in goldfish. So we can see that this data indicate that cerebellum-ablated goldfish are impaired in their ability to generate or use allocentric representation of the environment, and at first glance seem to support the hypothesis proposing that the cerebellum participate in integration of multisensory spatial-temporal cues in a cognitive map of the environment. The spatial deficit observed in the cerebellum-ablated goldfish, together with their ineffective searching pattern, agreed with the hypothesis and suggest that, as in mammal, the teleosteocerebellum is involved in adaptive sensoriomotor processing and consequently in the procedural aspect of navigation. Remarkable is the effect of cerebellum lesion in goldfish are profound role in spatial orientation and navigation, and it don't produce observable sensorimotor impairment or deficit in posture control, swimming ability, or obstacle avoidance. So, regarding all the data described before, we can summarize the following main conclusion. Comparative neurobiological research has shown that teleost fish can use a variety of navigational strategies that closely resemble those described in mammals and birds. Also see different brain circuits involve underline egocentrically reference spatial orientation in fish body center, and also another brain circuits that involve fish hippocampal pallium that is essential for encoding spatial information in map like relational representation of the environment that we know external world center, and is some of these similarities could indicate evolutionary convergence shaped by common environmental constraints and survival requirements. At least some of these strategies seem to be based on conserved neural substrate, likely shared with land vertebrates, suggesting that this strategy and their neurobiological basis could have appeared very early on during vertebrate evolution. So thank you for your attention and remain at your disposal if you have any question. Thank you.